All right, we'll go ahead and get started. Thanks for everyone for joining today. My name's Dean Horton. I'm the local representative for Nanostring here in Michigan. Um, Thanks again for having us here today. We're looking forward to talking today about Nanostring's spatial biology platforms. I'm going to introduce our technical sales manager, Seth Meyers. He's a technical sales manager for North America, and he'll be giving today's talk virtually. Nanostring spatial biology, transforming the information content obtained from a single tissue section. So thank you again, and let us know if you have any questions throughout. You can drop them in the chat. We can answer them there, and we'll take questions afterwards as well. So Seth, I'll let you take over.
Okay, great. Thanks, Dean. Quick sound check. Is my volume okay?
Yep, we can hear you.
Okay, perfect. So very grateful for the opportunity to talk with you all today. And as Dean mentioned, we're going to be covering our spatial biology platforms here at Nanostring. And the agenda we put together was first taking a brief look at our GeoMx Digital Spatial Profiler, which of course is available through the AGC. So we'll give a little bit of an overview around that platform, as well as a publication that came out as a result of the work at the AGC, looking at the analysis of donor pancreas tissue out of transcriptomic signatures in the microenvironment of early neoplastic lesions from Dr. Di Magliano and et al. So happy to provide an overview around that study. And then also want to give an overview around our applications around protein, where we've recently launched an immuno-oncology proteome-specific atlas assay as well. So happy to give an update around that specific assay. And next, we'll go into talking about our CosMx spatial molecular imager. So we'll give an overview really with this system and its assay. It's all about providing the most information per cell. So we'll give an overview around where we are by way of panel, a couple of case studies as well, and then a sneak peek into our 6,000 plex discovery panel, which of course is used for a really nice way of being able to profile across tissues at a 6,000 plex assay. So the highest of any commercially available assay that's currently available. As a quick reminder, right, these two different platforms we do find do provide complementary scientific questions for the use of their platforms. So when it comes to CosMx, right, this is all about helping us to understand how single cells compare with one another. And the way that we're finding the use of CosMx is this great tool that allows us to do it 1,000 plex today and in 6,000, and really providing the most information per cell. But to start with, we're going to talk a little bit about our GeoMx platform, which is this cellular enrichment application approach, where you're able to look at whole transcriptome across distinct tissue compartments, where it's really this hypothesis-driven approach of understanding and wanting to interrogate specific cell types across tissue. So when we're thinking about these two platforms, we always like to think about what are some of the different questions that can be asked of these two different approaches. So first on the right, as we're thinking in terms of our CosMx application, of course, because this is getting down to single cell resolution, really being able to understand how these cells communicate with one another. And then because of the plex that we have with our CosMx, really getting into understanding how pathways are impacted across different cells as well. So really looking at understanding things like differential expression across cell type A as an example, relative to its proximity to cell type B, or because of the 1,000 plex that we're at today, being able to look at many different ligand receptor pairings, and at 6,000 plex, over 200 different ligand receptor pairings, are really just looking to understand a basic tissue map across tissue. But to start, we're going to think about our GeoMx application. And with this platform, this is a great way of being able to not only understand specific genes, but their impacted pathways across these tissue sets. And as we're continuing to grow out our modules on GeoMx, being able to also look at protein is also providing that functional unit of measurement as well. So when we think about these two different platforms, just a high-level thought around some of these advantages that we have across these platforms. When it comes to our CosMx assay, being able to provide really the most information per cell at 6,000 genes, and being able to get into what we've found this really nice cell segmentation strategy, and not just relying on DAPI expansion is a nice tool in thinking in terms of CosMx. Then, of course, from a multi-omic perspective, being able to layer in protein readout as well as finding the RNA transcript readout across tissue sections as well. But to start, our geomics application, right, this is a really nice tool that allows us being able to provide high gene detection, being able to work across lots of different tissue types, including FFPE samples. And again, this multi-omic analysis approach of what we're building out with our systems and being able to look at both whole transcriptome and protein assays all across a single tissue section. So when we're thinking about our geomics application, again, we're offering whole transcriptome. It's a really nice biology-driven approach, this hypothesis-driven approach of looking at distinct cell populations and being able to work across FFPE, fresh frozen tissue, being able to work across different formats, such as multiple tissue sections, whole mount TMAs, and then being able to, again, both profile for RNA and protein. And because of geomics and we're harnessing the powers of next-gen sequencing, there's really, an unlimited amount of plex that we can get to using our geomics application. And when we're thinking about geomics, one of the nice features that we have is these adaptive optics that allow us to really fine-tune on specific tissue compartments and being able to look at profiling across distinct hypothesis-driven cell populations across these tissues. So when you're thinking about this geometric profiling approach, for example, being able to looking at these, how they distinct into these different tissue regions, or being able to do the segmentation profiling across distinct biological compartments within a specific region of interest, of being able to look at, for example, pan-CK positive and pan-CK negative cell types, all within that specific region of interest allows you to get a very rich data set of not only genes, but pathways as well. So one example that we always like to highlight when we're thinking about the value of being able to really look at specific cell populations is this example we have of looking at these islets of Langerhans looking across pancreas tissue. So within this particular study, we're able to segment to uncover interdigitated cell populations across alpha and beta cells within these islets of Langerhans. And not only are we able to understand the specific readout of these interdigitated cell populations, we're able to look at and really parse through these alpha and beta cells using these adaptive optics and select morphology markers. Not only are we getting imaging information, but we're also able to get differential expression out of these tissues as well. So you'll look between this pancreas tissue, looking at these biology-driven regions of interest selection across these alpha and beta cells that allows us to do this tissue masking across this compartmentalization that allows us to understand not only the genes, but we're getting into more of this pathway regulation across tissue molecular machinery. So this is a great way of being able to ask as a complementary tool to lots of different approaches. And where CosMx Connects come in is perhaps being able to array to define certain neighborhoods and looking at certain high throughput screening approaches as well. So one publication that came out here this past summer that was through the work that was performed there at the AGC. So this is looking across these donor pancreas that's looking at these transcriptomic signatures in the microenvironment of these early neoplastic lesions. So it's a great use case study of not only being able to profile across these CNR specific cells within pan-CK positive cells and negative cells, and being able to look across these tumor tissues as well, and looking at the poorly differentiated tumors as well as in the glandular tumors as well. So you'll notice here the overarching approach here is being able to generate this UMAP plot, which is essentially this overlay across the extracted epithelial cells from these single cell data sets across normal and tumor samples as well. So really nice use case study highlighted there through the AGC. Lots of different tissue types in research areas that we're finding that geomics is being utilized for. So here we're actually profiling across microglia, across 3 different neuron populations in the mouse amygdala. So here we're able to look at different classes of neurons, excitatory neurons, as well as the microglia, and being able to detect through our mouse whole transcriptome assay a number of different gene sets across user-defined areas of tissue that we've profiled for. But we're also finding when we're thinking about the geomics assay in being able to do whole transcriptome, and the cell typing assay with the promise of single cell resolution with the cosmics that are 1,000-plex provides really an unrivaled amount of data that's being generated across these distinct structures of tissue. So really looking and excited about the promise of what cosmics represents. So before I transition over to cosmics, I just wanted to highlight one of the latest new applications that we have. So of course, we have a number of different RNA-based assays, mouse and human for whole transcriptome, as well as a nice comparative oncology panel based on canine specific tissues. And then from a protein perspective, we built out these different modules that historically have run and profiled anywhere from about 40 to 96 up to 150 protein markers of your choosing across tissue. But we've next started partnering and continued partnership with companies like Abcam, where what if you could look at all IHC validated antibodies for immuno-oncology, and then you could compare these 570 protein immuno-oncology targets and add in some of your favorites as well, and then create essentially an IO spatial proteome atlas assay. And if you could combine that with whole transcriptome as well. So the drum roll, right? This is our official launch for the IO proteome atlas assay. This link, and we're happy to provide it following our talk here, actually highlights the specific content list we have for our IO proteome atlas assay. And there's a downloadable functional annotation listing that also shows all the clone ID information that's all included within this panel as well. So a lot of information and a lot of content that's being provided. When it comes to this specific application in this panel, we've partnered with Abcam. So all of the content that they've provided on their website is functionally validated. And so when we've created this panel, we then took a look and worked with other immuno-oncology subject matter experts, specifically working across CTAP data sets. And we've been able to create this specific panel that we find is the most valuable for any immuno-oncology researcher that looks across a broad array of post-translationally modified protein targets. So this panel is specific to human, and the protein content has been mapped bioinformatically to 556 unique specific genes of interest. We're happy to provide that gene listing if that's something that is of interest. These are functionally validated antibodies that we've gone through. Again, we partner with Abcam. And so what we've done is we've essentially created a 90 pellet CPA, as well as testing across an FDA-approved TMA across tumor and normal tissues. And then we found that this is really a nice way of being able to specifically target with great sensitivity and no evidence of antibody interference at a 570-plex panel assay. So one of the first data sets that we took a look at was looking specifically at liver cancer. And so here we're looking at pan-CK positive cells, CD45 cells as well. And what you'll notice here is that we're actually able to look at the tumor side as well as the immune side very quickly across tissue, and being able to then understand the specific proteins of interest that are present across tissue as well. We've had a couple different studies that we've performed. So the first that was performed, this was provided at the Society for Immunotherapy conference this past fall, where we actually worked with Mayo Clinic, and we're able to use IPA looking at HER2-positive breast cancer tumor biopsies, looking at both responders and non-responders to neoadjuvant treatment, and were analyzed then with IPA. And the whole goal of what this panel represents is the ability of describing and finding and uncovering these new biomarkers of interest across a single tissue slice with one aliquot of these 570 protein markers. So a really quick and easy way of being able to perform this type of analysis. There was also a really interesting data set where we actually looked to show the use of the Cosmics 1,000-plex RNA panel as well. And we saw a really nice concordance again, because we've recapitulated those 556 RNA markers that are present and correspond to these 570 protein markers as well. So that's a really interesting poster that came out this fall. There's also a nice study that was done out of David Ting's lab out of MGH, where they were actually had been big users of RNA sequencing, but had wanted to be able to look across these pancreas specific samples and looking at a number of different markers with protein in this case. And what they found was that they were able to do both IPA, the 570-plex protein assay, and whole transcriptome. And that was really highlighted this immune response of short versus long survivors after combinatory therapy. And they were able to find these differences that were small in the spatial RNA data, but more dramatic within the spatial protein data as well. So really interesting data sets we're getting across a number of different tissue types as well. Here's also a proteogenomic application specific to CD44, where we actually found that that CD44 was significant at an RNA level, but not translated at the protein level. And so being able to look at these specific glioblastoma, multiform immune infiltrates at both the RNA and the protein level were shown to be very impactful in being able to look at these specific markers. So we've done a couple different studies here. This is also showing the proteogenomics across RNA as well as protein on the same tissue section. And the workflow that we have is a very comparable one in being able to profile across these. And so you'll notice here when we're able to image for using our geometric region of interest profiling and using segmentation specific profiling, you're getting a nice rich data set across CD45 and in this case GFAP markers that were important within this particular study looking at glioblastoma as well. So nice data sets were being generated across that, and that's the specific case study publication that referenced our proteogenomic specific application and their workflow. And again, we've highlighted that workflow in being able to work with both protein as well as RNA. And here we're highlighting within CRC-FFP specific samples as well. So a nice biomarker, real discovery type of an approach that you're getting across tissue with our IO Proteome Atlas assay. So when we're thinking about genomics, right, there's over 330, I think we're at 336 peer-reviewed publications across what we like to describe as real-world samples. So exciting times that we're finding across genomics and their use around real hypothesis-driven applications, and specifically thinking about the genomics whole transcriptome and now the IO Proteome 570-plex protein assay as well. So next, I wanted to transition over to our CosMx spatial molecular imager. So I wanted to give a couple different examples of some of these case studies that we talked about, as well as a sneak peek into some of the new information and new data sets we have generating our 6,000-plex panel. So as we were thinking about what CosMx represents within the spatial imaging market, what we like to think about, again, is providing the most information per cell. And so at that 6,000 plex and increasing, we also have, and we'll demonstrate here, what we describe as a really nice cell segmentation strategy that's based on a multimodal approach. This is available today and how we're actually running the assays on CosMx. We also have the ability of providing RNA and protein measurements. And we also like to look at our tissues, similar how we've built out our genomics application and being able to work with not only FFPE samples, but being able to work across fresh frozen and then different formats as well, because you're using your own glass slide, the ability of being able to look at TMAs, multiple tissue sections across tissue. So really nice use of the platform from that perspective as well. And we've also looked to use the same workflows that we've generated using our genomics, so standard IHC applications, known proven applications that can be used for being able to set up assays and running across tissue as well. When we're looking at the genomics and the CosMx system, they do share an in situ chemistry, although they may be different in the readout, right? Knowing that the CosMx platform is an in situ hybridization chemistry, everything happening on the CosMx system. But what we are describing here is the ability of having a really highly multiplexed assays because of these in situ chemistries that we've developed using these platforms. And when we think about CosMx, again, we are designing our assays to provide the highest plex and high resolution with our platforms. The probe and fluorophore design really helps to reduce optical crowding limitations on plex, similar to what we've experienced with genomics. We don't run into those saturation issues using our platform. We've fully achieved our 6,000 plex assay. We have a Nature publication that actually highlights the use of the platform for that perspective. And we have publicly available data sets that demonstrate our ability of performing that 6,000 plex assay. We're also in terms of customization and thinking about being able to design assays for things like chemokines and cytokines, the ability of targeting less than 1 kb in length as far as transcripts go. We find that there's no loss in performance, and the ability of being able to actually design gene sets for those types of transcripts as well. We've also found this system to perform equally well within FFPE as well as fresh frozen tissues as well. We do have an upcoming webinar that is happening tomorrow. Of course, there's the ability of being able to replay this on demand, but it's a publication that's coming out that resulted from the Mayo Institute as well as the Buck Institute that's specific to senescence. So for those that might have an interest around some of these neuroscience applications, wanted to be able to highlight the specific webinar that's coming out that's being performed by Dr. Marissa Schaefer out of Mayo Clinic, Rochester. So interesting applications that we're seeing, in this case, using our mouse 1000-plex neuroscience RNA panel on CosMx. And there also is a use of our geomics application for the mouse whole transcriptome as well. So really exciting to see some of these different applications that are being used together. And in this case, for the CosMx perspective, right, able to profile these specific senescence markers with the generation of this ligand clustering heat map, and also looking at the development of these top differential expression genes as well. So really exciting data set that we're happy to have a researcher provide their insight into the use of the platforms, and more importantly, the science that you're generating using these platforms too. So as a quick reminder, when we're thinking about CosMx, this is an in situ hybridization chemistry and readout. We'll talk a little bit about our sample preparation and thinking about the hybridization of your specific RNA probes and panels, or the protein-specific antibodies that we're using. Again, we are compatible with FFPE as well as fresh frozen tissues. And then after the sample preparation occurs and getting that flow cell assembly assigned to that particular slide, you're loading that onto the instrument, and that's where the integrated readout, that imaging as well as that cycling quantification is occurring on instrument. And following that specific readout, we are uploading our data into our Atomic Spatial Informatics platform, which is a great tool to being able to not only to store data, but to share data. And there's some nice modules that are being built out for Atomics as well. So excited to see that portion of the workflow coming along as well. When we're thinking about the tissue formats, right, have your sectioning your tissue, just like we're experiencing with our geomics application, being able to load here single tissue sections, multiple tissue sections. Here we're looking 6 mouse brains that are loaded onto a slide, or a TMA. There's really a wide variety of format that you can take in being able to run on the CosMx platform. In terms of our sample preparation, we've really looked to being able to mirror a lot of the work that we've done with geomics and some of the lessons learned in thinking about the geomics application, in not only being able to do a manual preparation of your slides, which has about a 2-day overarching workflow time with about 2 hours of hands-on time. We do not require tissue clearing or hydrogel embedding, which you can experience with other platforms. But we've also created a semi-automated workflow in terms of the staining with the use of a Leica Bond RX system. And this is the same system that could be used with both the geomics as well as our CosMx platforms. That helps to reduce hands-on time to about 30 minutes in terms of the help that the auto stainer makes, in this case, helping towards cell segmentation markers that are used there. So I always like to give a ground set on some of the different assays that we do have available. So as we're thinking about the different assays and panels that we have a 64-plex immuno-oncology human protein panel that we have. There is the ability of adding on 8 protein markers of your choosing into any of these protein panels. The second protein panel that we do have is a mouse neuroscience specific panel as well. And all these are functionally validated in being able to order the vial, and then you get the panel the next day, and then you can profile your tissues on these protein assays. We also have, of course, our RNA assays. So we have our human universal cell characterization panel at 1000-plex. I'll share a little bit around some details on that panel. And then we've also created a recapitulation of that human panel in this case for mouse as well at 1000-plex. And then we do have a specific mouse neuroscience 1000-plex panel available as well. And then we have our human 6000 discovery plex panel again at 6000-plex that is coming out and will be officially available running locally here Q2 of next year when that'll be available to run. We do have some custom, as I mentioned, for our protein assays, customization as well. You can create some custom assays of your choosing. You can spike in for our 1000-plex panel, 50 genes, or for our 6000-plex panel, you can spike in another 200 genes of your choosing into that specific panel. So when it comes to first our 1000-plex panel, so this was the first launch that we had. This is a panel that's readily available today, both for human and mouse. It provides, again, the same accurate multimodal cell segmentation strategy, which we'll share. It's compatible across a wide range of tissues. And then in terms of its analysis and output, we're looking at being able to perform not only a really nice, robust cell typing application, because at 1000-plex, we're finding that we're able to profile and perform this cell typing in a very rich data set, but we're also able to then also profile for both cell states and interactions, which perhaps might be one of the more differentiators as you're thinking about CosMx and how it represents and how it's different within the market. So again, this is a panel that's available for both human and mouse. And when it comes to this 1000-plex, here we're demonstrating the mouse universal cell characterization with the coverage that we're finding across these specific panels as is assayed across tissues, looking at hormone activity, getting into cell-to-cell interaction studies, signaling pathway and target genes, looking at cell state and function targets, and then cell typing as well, kind of rounds out the use of the specific panel. This is one study example looking at the mouse universal cell characterization panel. The link is there. It's available to be downloaded. So this is actually looking at immune-mediated control of lung adenocarcinoma. And there was a nice comparison of being able to compare to single-cell RNA sequencing mouse lung data sets that were developed as well. And of course, what you're gaining out of CosMx is not only the quantification of these specific genes, but you're also placing a home of the particular cells within tissue and then the genes within those cells. And so this is a really nice CosMx cell type map that's performed on the basis of this 1000-plex panel as well. So next, I'd like to talk just a little bit about how we go about in performing our cell segmentation. So this is looking at this multimodal approach. So this is based on a couple different iterations that allow us to perform this multimodal cell segmentation approach. So there is a image pre-processing step that's taken. There's machine learning cell segmentation approach that we've been utilizing for about 1.5 years now as well. So that continues to influence and help us with our cell segmentation. But what we're doing here is we're not only utilizing protein nucleic acid imaging-based cell segmentation. So we use a couple different ways of being able to image this data. It's not just a DAPI expansion that we're utilizing. We have 5 channels that we utilize in addition to just a DAPI expansion. You're able to use some morphology markers specific. For example, there's a cocktail that we use, beta-2 microglobulin and CD298. And there's also a couple different markers we can use as well that help to influence cell segmentation as well. So really nice approach that we have. And that's all about helping us to being able to assign the transcript in its correct home, in its physical home within the cell. And I'll show the impact of what that makes across tissue. So here's just a couple of examples we've seen before. So this is across a variety of different tissues. And I'll just highlight here that for the example for mouse brain and fresh frozen, right, we're utilizing ribosomal RNA, GFAP. So a couple of different markers that we have for cell segmentation. And then dependent on different tissue types that we're using, for example, if we're working with colon tissue, being able to work with pan-CK is informative as well across tissue as well. Here's a more recent new set looking specific at neurosciences. And in this case, because we have these 4 dedicated protein markers that really help us to derive these advanced cell segmentation markers. So looking at these astrocytes, these microglia, being able to really look at these specific neurons as well. That the shape and borders of these particular cells is something that the advanced cell segmentation is really noted across using CosMx as well. And here's another example of being able to use this ligand receptor pairs, in this case with our 6,000 plex panel, where we're looking at uncovering the correlations of some of these different markers and specific neurons as well. And so in this case, we're actually able to look at these really rare cell types that are identified because we're using these 6,000 genes that help us to further inform and perform this cell segmentation as well. So it really helps this really rare cell typing at single cell resolution that you're getting using the 6,000 plex panel. So when we think about the advanced cell segmentation algorithms that we use, we're also incorporating some nice open source tools as well. So when we think about from an RNA perspective, we use a modified version of CellPose. And when we're thinking about protein, we have a modified version of Celesta. And that's a really nice tool that we're going to continue to develop and use in being able to do and further enhancement to our cell segmentation. So here's just a quick example of the impact that cell segmentation has on our data sets. So as you'll notice here, because we're not just using a DAPI expansion, this boundary here in the dark navy is if you are using something like a DAPI expansion. And what you'll notice here is the red circles are a specific transcript that's being misassigned within this particular cell. And so what we're able to do is actually create this cell segmentation in this green that allows us to being able to call that specific transcript of interest and assigning it to its correct home. And what we're finding is that these minor cell segmentation errors, they could lead to transcript misassignments, and that impacts everything downstream, right? Being able to introducing a downstream bias into this data analysis, including clustering, differential expression, and pathway analysis. So what CosMx is able to provide is being able to do this multimodal cell segmentation strategy, utilizing cell morphology and not just relying on specific DAPI-only cell segmentation. With just a DAPI expansion, you're prone to having transcript misassignment, and that can impact your data sets. This is just showing a publication that's coming out that's from the University of Tokyo. Similar tissue types. This is with breast cancer specific tissue, and this is just showing a visual comparison across cell segmentation with those that are using this multimodal approach, which you use on the left versus just using a DAPI expansion and segmentation. And really the impact there in the final quadrant is just showing the impacted size of those cells that are matched to tissue areas when they're compared. And there's an impact that's made there downstream into the analysis as well. So this is showing the in situ type cell typing algorithm that we use within our application. So this provides 3 modes of cell typing. So getting into supervised, semi-supervised, and completely unsupervised. This is a bioRxiv paper that's out that we developed in concert with the launch of the CosMx system. And this is also showing the promise of multiomics, running a serial section. This is looking at a core needle biopsy of being able to profile our 64-plex protein assay, which I'll share the content on the next slide, and being able to, in this case, using our 1,000-plex panel as well. So really looking at these ligand receptor pairings that show the expression level of these specific tumor cells across tissue as well. This is the 64-plex human immuno-oncology protein panel we have read out on our CosMx system. So as you can imagine, a lot of content within the immune, the cancer, the stroma content. Each one of these have all been functionally validated and being able to work together in a really highly multiplexed fashion as well. And here, this morphology line shows you some of these different segmentation cocktails and morphology markers you can use in studying immuno-oncology as well. So the value of plex that we're finding, right? This is looking at if you're just measuring something like 280 genes at a time. It's a Veroni plot where the darker the green, the pathway, the more coverage of that specific content. And as you're getting into a 1,000-plex assay, what you're really getting into is this nice comprehensive coverage across many of the different pathways that you can find across what we consider a kind of a universal cell characterization approach. And so with that, as we highlighted before, at 1,000-plex, you're able to get many of these different analyses within one specific experiment. So in being able to do a cell atlasing, the neighborhood analysis, again, getting into cell state and function, our ligand receptor interactions that are paired automatically within these panels. In this case, we're looking at our mouse neuroscience panel, being able to look at these microglia and oligodendrocyte pairs, and then finally getting into this pathway analysis as well. And as we're demonstrating across different panels and different gene sets that are out there, the benefit of being able to look at this 1000-plex, in this case, we're looking at a 950-plex assay. We're getting into more of this understanding of differential expression, right? In this case, how do microglia near tau plaques change behavior, or how do T cells change near tumors? How do they change expression? We're getting into more of this pathway analysis of these different changes as well. And finally getting into kind of these ligand receptor pairs as well as how they're physically interacting as well. So a lot of rich, meaningful data that you're getting from this. So next, I wanted to just talk a little bit about the 6,000-plex assay that we have that's coming out now. So again, quick reminder from an RNA perspective, we've got our 1000-plex human and mouse universal cell characterization panels. We've got our 1000-plex mouse neuroscience panel, and then we've got our 6,000-plex human discovery panel as well. And so for this specific panel, it's got over 6,100 curated RNA targets. You can customize and adding an additional 200 targets of your choosing into the panel. We've got the nice same accurate multimodal cell segmentation approach that we demonstrated with our 1000-plex panel. It is widely compatible with a wide range of different tissue types. So I'll highlight a publicly available data set that we have specific to human brain tissue. There's also some a data set that I'll share that looks at skin as well. And the nice thing about this 6,000-plex is really getting into being able to profile nearly the entire human reactome. So it's a really exciting level up in terms of the plex and the amount of information, again, that you're able to generate across tissue. So this is a publicly available data set that we have, the first that we've launched here. So this is looking at the human frontal cortex. And from this, you can access this data set, you can download it, and you can actually take the imaging information that's generated. You can also take a look at some of the data files that are generated as well. There's XY coordinates, just flat files of the genes that are present within tissue, the cells that are identified through a cell type map, and a really nice way of being able to do this UMAP projection of cell clusters as well across the identified cell types that are present there. So that's publicly available. And then this is a study that was done, again, using the 6,000-plex panel that was done at the Society for Immunotherapy here last month. And what this does a really nice job of is what we're highlighting here is the discovery of different cell types, cell states, and then understanding how the cells are responding to their environment. So like the overarching cell responsiveness. And what we found from this particular study, again, looking at skin cancer, was that we found, CosMx enables the ability of looking at spatial measurements of these 6,000 genes simultaneously. We've used a protein co-detection of a single achieved FFPE tissue that has really nice high efficiency as well as low noise ratio. And then with the panel, we found that we're able to profile 1 million cells across multiplex tissue sections within days. And this is a tool that really provides this novel tool to reveal what is the spatial signature, in this case, of the tumor microenvironment and the oncogenic pathways. And that can really help us to really look at that next level of cancer and therapeutic research. So really happy to see this poster that came out of SCSAP, and just taking a little bit more of a look at some of the different data that was generated specifically from this specific project. We're looking at these cancerous cells that are subclustering into 7 distinct spatial populations. And here are the spatial transcriptomic. This really allows for the visualization of these novel cell type patterns in this really, really complex tissue microenvironment. So there's kind of this rainbow effect of the cell type map because we're looking and identifying so many of these across tissue. And with these cell typing that CosMx enables, we're able to confirm the spatial distribution of not only the cell types with the help of these corresponding marker genes that are most differentiated within these groups of cells. And what we find is the alignment of these protein stains as well. So this figure, what this shows is the co-localization of specific cell types, the marker genes, and then the stained proteins for these 3 major cell types in this cancerous skin region. So the top rows are these morphology staining for plasma cells, for CD45, keratinocytes, PNCK, and then macrophages in that teal for CD68. And then here in this middle row, we've got these cell type marker genes for plasma cells, and then for keratinocytes, and then for macrophages as well. And then within this bottom row, we're looking at the cell typing results shown within the CosMx using the in situ type algorithm that we're employing within every run of our application as well. So this cell typing that we found, it showed many of these different cancerous subpopulations, which revealed these spatially from cells that lack really any defining marker genes. So what we found was that we matched the structure of these keratinous pearls, and then we identified through an H&E stain, so for further verification. So these visual inspection, it shows the cancer populations revealed a concentric rings, which is underscored by visualizing the most distinct marker gene from each cancer cluster as well. This is taking a look at the use of our one of our latest applications, because at 6,000 plex, we've developed this in situ core. And what in situ core does is it helps to identify these gene hotspots through organizing these normalized values of gene expression across cells nearest to these 50 neighbors. And then we kind of group them spatially within correlated genes as well. So this hotspot, it shows an uptick in the genes that are responsible for antigen presentation, lymphocyte chemotaxis, and then cytotoxicity across these cluster of keratinocytes as well. So really, really nice way of being able to look at non-trivial specific markers in gene correlations. So from this, we were able to look at these macrophages that were defined as the near cancer of at least one of the 10 nearest cells that were annotated with a cancer label from cell typing. And then the average gene profiles of these near cancer, far from cancer macrophages were compared. And along with these profiles of these near cancer macrophages and cancerous, it really looked to eliminate this bias from a segmentation error. And so ApoC1, OLR1, SDS were all identified by finding these intersections of targets that really favor these macrophages near cancer relative to both the profiles, and that were confirmed with the spatial plotting as well. So really interesting data set that we've generated. Again, this is the 6,000 plex across human skin cancer. Feel free to download the poster as well. We'll provide the link for in being able to profile as well. So I just like to just kind of think about what are some of the interesting questions that we're finding with our 6,000 plex panel. Like how are these assays being used, and what are some of the great ways and great questions that folks are able to run these assays. And what we're really fundamentally finding is because we're offering the highest plex, getting into differential expression across cell type A close to cell type B, getting into the differential expression of single cells that have a specific biomarker, because it's at 6,000 plex, really getting into uncovering these biological pathways and how are they changing across space, across a single tissue section. And then getting into spatial context again of these ligand receptor pairings, because we're able to profile over 200 of these that are already preset and pre-built into the panels. And really just what are these new, great, exciting new spatial biomarkers that we could have missed using other techniques as well, because we're getting into 100% cellularity with these type of assays. It's exciting. So when we're fundamentally, what you're really looking at from the CosMx perspective, what you're able to get, again, being able to do something like a serial section, is really getting into this multiomic analyses, right? So at 6,000 plex, being able to look at tissue and being able to understand within a particular, what we describe as a field of view, you can then look at the single cell, what are the cells that are present within that field of view, and then understand the genes that are present within those cells. And at 6,000 plex, which is the amount of information that's generated, is a very rich data set. And then being able to layer in protein analysis with morphology markers and custom targets for quantification just provides a really rich data set that you're able to generate. And so with our 6,000 plex panel, we're really excited about the amount of discoveries and insights that can be made using this type of an approach, and the amount of content that's generated there across pathways. And then I'll just conclude with, there's a number of nice publications and preprints. We're tracking at about 40 that we've seen across using CosMx, anything from using our own publicly available data sets to further inform and create really nice new tools for spatial analysis, to working across a variety of different tissue types as well, using the application. So really excited to see the new publications that are coming out and leveraging this type of an approach. It's really a very exciting time. So I'll pause there and ask if there's questions or things we wanted to talk more about, but very appreciative of the time today.
Great, thanks, Seth. Do we have any questions? Either those joining virtually or those here? All right, Seth, we had a question of whether the 64-plex protein assay can be overlaid on top of the 1,000-plex assay.
Yeah, the way that we're envisioning running the assay today is on a serial section. So the way we would look today is to do a serial section of the RNA and the protein. With these systems that are coming out and being used locally, there's some interesting applications that are being used, and perhaps running the assay and then doing a xylene application in between, and then being able to run the 1,000-plex following that. But the initial data sets that we have that are being run are on a serial tissue section. But we have demonstrated some proof of principle of being able to do just that, of layering the 64 protein assay in the 1,000-plex panel as well.
All right, I think that's all the questions we have. Thank you, everyone, for joining us. There's extra pie in the back if you want to take some pie with you. And I have a card back there. Again, my name's Dean Horton. I'm your local Nanostring rep. And thank you again, Seth. And thanks for all who joined us virtually and in person. And great, feel free to email us with any questions. Thank you.